d'abord, je voudrais dire quelques mots en français.
First of all, I would like to say something in French.
Je pense au nom de toute l'Assemblée remercier les organisateurs pour
The entire Assembly would like to thank the organizers for the past 2 days. It's been fantastic and the organization was great. And I would also like to reiterate how important Françoise is. We owe her a lot. The community owes her so much. She has so much energy, she has so much endurance and stamina, and she's so elegant. It's the elegance with which she's inspired people in the past. And how we can turn this into a project or a study. There's still many things we don't know yet, but the sustainability and the female endurance and stamina, her perseverance, that's fantastic. And I think we really have to thank her for everything she's done.
Le deuxième point qui lui aussi est féminin, c'est
the second point, which is also female, I would like to thank Magalie Viallon. She's my partner in science, but also in life. And just like with Yvon Le Mao, we were introduced into the life of fasting, and it's this female sensitivity that has made scientists study this particular area. So I would like to thank also my wife and Françoise again.
I will now switch to English to make the presentation. Here are my disclosures. And the first question I think we need to ask, and you probably ask yourself, is why are we discussing about magnetic resonance and how it could help in fasting intervention research studies. The first point I would like to make, and that's probably a good argument, but not the only one, is that MR is frequently the method of choice for pharmaceutical companies, and that whenever they want to demonstrate the efficiency or the added value of their new drug, they do use MR to obtain their approval from the authorities. And one of the reasons is because it has usually an increased power in the study design, so it means also less money that spent to demonstrate and to reach the goal of the study. And just an example of one of the studies that have been showing that whenever you're using MR compared to ultrasound, you can dramatically decrease the sample size to have a given effect. Okay, so it means that it is more efficient than other imaging methods just because it provides reliable and quantitative measures. But there are more. And you can see here you have the whole body picture. It's not me, I don't know who it is, but it's usually used as a reference method for anatomical or phenotyping studies, whether it's small animals or humans. Second, and it's a big advantage, it's a non-invasive and radiation-free imaging technique that can therefore be repeated as many times as you want. This is very important for follow-up studies, especially if you do it on volunteers and people that are well-being. But there is more. It provides not only anatomical information, but it also provides many quantitative and functional markers and measures. Here is an example of what we did on a longitudinal study in ultramarathon studies, showing that there are changes in the water content in the brain. You can study the content of fat in the abdomen and the liver. This is what we'll be discussing later on today. You can look at what's going on in the heart as well, water, fat, whatever. And you can also do biochemistry, and we'll be discussing very quickly what are these spectra. but you can do images, spectra, biochemistry with the same tool. Okay, it's a big tool, but I will explain it shortly in a few seconds how it works. So the idea here is to make the point that we are moving or trying to move from global markers, blood samples are global markers. When you get the sample to measure the fat content in the blood, it reflects what's going on in the whole body, not in a localized fashion. MR opens the scope of providing local markers in every single pixel of this image. I can know the number and the value. It's like multiple biopsies performed non-invasively that can be repeated over time and over and over. So it's really the power of it. In a few slides, I would like to explain you what is MR, MRI and MR spectroscopy. So I'm not sure you will be a senior physicist at the end of these 10 slides, but at least I hope you will gain some knowledge on how it works and what are the basics. So MRI is, as you know, meaning magnetic resonance imaging. It can be spectroscopy, as we will see. And in 95 or 99% of the case, we're assessing and looking at water molecules. And you need to remember that water molecules, it's an oxygen atom and 2 atoms of hydrogen. And this hydrogen nucleus, the proton, it can be considered as a very small magnet, okay? So because the body is essentially made of water, you are made of many, many, many small magnets, and this is actually what is the matter we are looking at. So if you are sitting in the room, of course you're not magnetized, okay? So if I'm looking at your small magnets, they're in all random direction, and finally your net magnetization is 0, hopefully, because otherwise there will be conflict in the room, okay? So the thing is, whenever you put a body in a magnetic field, such as MRI, what happens, depending on the field of the magnetic field, it can be, usually most of the scanners are 1.5 Tesla, that's the most common ones. We are now having for high-end clinical scanners using 3 Tesla scanners, and it can go up for research, very experimental study with 7, 9, and even there is even 17 Tesla scanner. But most of the case in humans, we're using 1.5 or 3T. The point I would like to make is whenever you're using such magnet, what happens is that you're reorganizing these magnets. And you can see here, if you're applying to these spins, there is a difference between the 2 energy levels, and this will be what you will be looking at. There is a small difference between the 2 energy, and this will be the signal, the source of the signal that you will get. And the second point I would like to make is, when you are in the magnet, if you're using a stronger magnet, you will get higher signal. And that's explained why for research studies we are most of the time going to 3 Tesla, because we get more signal, so it means more speed or different, whenever you have small signal to noise, more power to study these details. So you can summarize it like this. If I take a subject, if I don't put it in the scanner, there is no magnetization of the tissue. Within the scanner, I create some net magnetization from this difference between the 2 energy levels. So we are prepared to make an experience, MR experience. But the problem is that, this is just to show you a different, which scale factor we are talking about. This is the strength of this magnetic field of the proton. You have the Earth magnetic field, which is 50 times higher. And here you have an idea of how powerful the the magnetic field is whenever you're talking about a 3 Tesla scanner. And what happens is if you just put the patient or subject in the magnet, what happens is there is no way to distinguish this very small vector from the big vector field in the scanner. They are mixed together, and we need to do something more, okay? So the something more we're doing is actually we're playing music. We're actually using radio frequency and sending to the body a given music partition, and we're actually transmitting at the proper frequency a given amount of energy to tilt this vector, this net magnetization of the body in a given voxel. So you get it in the transverse plane, okay? Just a little mathematic or actually representation that we probably learn in the 2nd grade. So we are representing the vector as 2 components, the transverse plane and the longitudinal components. So these 2 components are one of the basics of what we're looking in MR. And what happens whenever you stop the music, what happens is that this vector return to its baseline position. And you can see in these 2 animations, this component, the longitudinal component, increase, and the transverse relaxation, the T2, it actually decreases. And these 2 parameters are the basics of MR that are 2 independent features that characterize water in the different tissue, and that we'll be using to make the nice, colorful images that we'll see later on in the presentation. So keep this in mind, and so from that standpoint, we can obtain different contrast. It can be grayscale, it can be color scale, whatever. What we can see is that there are different behavior in this T1 and T2 relaxation that can help us to distinguish fat in the subcutaneous compartment from muscle or from blood, okay? So it allows us to build up a contrast. And T2 weighting is another dimension. It means the interaction with other molecules, and it's typically a brain image where you have the T1 weighting, T2 weighting. Here is just the amount of water, and you can do it, of course, in every organs, okay? So that's the basic principle of what MR is doing, measuring after putting it in a magnet, a constant magnet. You have this relaxation experience, and we are repeating this music, so we're not just playing one sound, but it's a real partition. It's like an orchestra partition we're playing to build up the images we'll see later on. Okay, so just a take-home message for MR imaging. I would like to remember and to understand that the imaging signal, what we're measuring comes from the water protons and its properties, T1 and T2 relaxation. It is very important. You can measure, and you can measure it and to differentiate tissue. There is something else. It's called MR spectroscopy. MR spectroscopy is something like is biochemistry. In the 70s or even the 50s, where people have been discussing about NMR and not imaging, but nuclear magnetic resonance to do biochemistry in samples to know the composition of this tissue. And this is what we can do in vivo in patients or in subject. And so we actually taking some sample, usually it's voxel of bigger size, and in this voxel we are measuring protons that come from other molecules. Of course, we have the one coming from water, but usually we want to avoid water to see what are the different molecules within this sample. So that's the different components that contain proton in the case of whatever it's MR spectroscopy of the proton. And a very important point to understand is that we are looking at concentration that are, of course, much lower, where it's a matter of 1,000 over 1,000. So we have low signal, usually it's more complex but we have a unique information in terms of composition. So we can do it on proton, the H1 nucleus, but you can do it on other species. And this is really where the power is. You can study phosphorus, nitrogen, carbon, fluor, and many others. That's the most common ones. And so what it means is that actually we're looking at components in the tissue that contains, for example, carbon atoms, so, such as glycogen. So you can sample the glycogen in the tissue. If we are looking at phosphorus, and we'll be discussing this later on, you can measure and quantify the amount of different variants of ATP, phosphocreatine, phosphate, and the pH is actually measurable in vivo. So it's a really powerful tool to assess what's going on in the different tissues. And last but not least is one of the strengths of MR spectroscopy and phosphorus spectroscopy is you can do it dynamically. Here you can see an example where you have someone contracting a quadriceps muscle, and we can do it in each individual muscle to actually look at the dynamics of this spectrum over time during contraction. So you may set up and tune the strengths needed to actually explore dynamically what's going on in terms of muscular contraction. And in here you can see this phosphocreatine peak going down and just re-increasing. And you can see it here in this graph. So it's briefly the same thing, but on this side. And this graph is really what makes its strength whenever you want to look at oxidative metabolism, especially because here you can see the different section of this graph. First, it assess the ATPase rate and somehow the power of the contraction people can do, and then you reach a steady state. So it means the work-cost relationship, and then probably the most interesting thing that people currently are interested, or at least we are interested in, the recovery rate. This recovery rate, the tau constant that can be calculated from this recovery, is really interesting because it means measuring the oxidative metabolism in the mitochondria. So it's a real powerful tool that is usually used by people looking at exercise and sports research is really using this tool to explore the oxidative metabolism. And I think this is typically the one thing that have to be performed to increase our knowledge in the understanding of what the fasting is doing to our body. So now I will be switching to the study we've been performing. It's some kind of unusual study. It's actually it's a feasibility study. We came here in Berhinger to see and to do a preliminary study to see if it is feasible and what are the potential of some of these techniques to explore, measure, quantify the effects, the immediate effect and the long-term effect of 14 days periodic fasting, and this being performed in multiple organs. Okay, that's one of the strengths. You cannot just focus on one organ, but you can explore different organs altogether. So, of course, it's feasibility, more or less a pipeline assessment of what we would like to perform in a larger study. But I would like to share this paper that was published in March. And that was performed, of course, with Françoise here in Berhinger. So that was a RB-approved study. As a case report, is one subject. There was a 15 days Bringer periodic fasting intervention in the Überlingen clinic, so that was the one way to do it. So we were sure that that was done properly with 7 days pre-fasting vegetarian and 14 days stepwise refeeding. And this longitudinal study was performed with MR imaging and biology samples taken at 4 different time points. That was performed before the fasting, at the end of fasting, before refeeding, and then at 7 days, 1 month, and 4 months, knowing that at 1 month we focus on muscle and 4 months on the abdomen data. And we looked at, as we'll see, high and low abdomen and tight level for the characterization with MRI and MR spectroscopy. So here are some details regarding the methodology that we applied here. So we applied in the upper and lower abdomen level a given acquisition protocol, which is something quite, I would say, basic. It's called a 3D Dixon acquisition. So this is the imaging, but what the beauty or the strength of this acquisition is in the analysis pipeline that is based in several papers that are based on the work of one person in our lab, Benjamin Leport, that has been working hard in the quantification of fatty acids in different organs and in normal or different phenotypes. And based on this subject, we have the capability to quantify not only the amount of fat as a whole, but also the fatty acid composition, as I will detail in the next slide. So here you have the same graphic, but larger scale. And from this data, we apply this model-based quantification after automatic segmentation of the subcutaneous and visceral abdominal tissue, fat tissue. And we are, this is some intermediate result where especially we have the fat fraction here. And then from the different characteristics that are the chain lengths and the type of bound and in the kind of bounds, we can differentiate the saturated, monounsaturated, and polyunsaturated fatty acids. So we have the capability to extract from the spectrum the different types of fatty acid that are in a given voxel of the tissue and every slices of the body that we imaged. So fatty acid composition is an interesting feature results, and of course since we're doing automatic segmentation, we can calculate the visceral adipose tissue and the subcutaneous adipose tissue. Of course, we can do other things, especially when you are considering bones. When you are considering bones, we can measure the bone marrow adipose tissue as well. And we can look at also, so there was supposedly a movie here, but this was to show you the segmentation models that were deploying in that kind of study that were developed for other studies, but all other needs for longitudinal extraction of the data. And actually, that was done in the same study, the MUST study, where we were looking at longitudinal changes on the muscle and the volume of the muscle. And we are using some deep learning algorithm to perform that in this subject. So you can see that it's not just simple images, but it's different models that are put in an analysis pipeline that allow you to extract numbers that are numbers of interest, the composition on the fatty acid and the different volumes of organs of interest here, the muscle, the fat, as well as the bone marrow. And remember that we have bone marrow both in the spine, that was in the, I will give you results on that, and of course in the bone of the thigh. Okay, so that's, I wanted to show you how it works. I mean, this is actually when it works just like this. So this is the strengths of this algorithm that are the capability just to look and that have learned on database to do the segmentation, apply a given template on the muscle to finally end up to a very accurate way to extract each individual heads of the quadriceps muscle. Here I will just give you a result on the sum of each of the heads of the quadriceps muscle, but that it can be of interest potentially to look at potential differences in each individual heads of the quadriceps muscle. Nevertheless, here are some results. So most of them in this slide are in line to what, and it's not a surprise, are in line with the study that was published this year by Françoise, showing that after 14 days the weight loss was near 8 kilograms. 4 months later, at the last time point, it was 5 kilograms. There were changes in the biologic profile, the overall profile with what is known there, of course, lowering of each of the components of the both fatty acids and the triglycerides, and more complex molecules of the lipids, including triglycerides, LDL, HDL, and so on. Okay, so this is known. Maybe a focus on the liver enzyme that as well increased, and showing dropping down. 4 months later, it was at the same or little below, but I think it's part of the statistic fluctuation. Nearly the same for SGPT, but SGOT was a bit higher. And of course, this is known that all the acid uric and urea are changed and with an increase, and that was well described yesterday. And the leukocytes as well were decreased after this 14 days period of time. So now let's look at the different stages, and I would like to focus first on the lower abdomen adipose tissue analysis. Here you see here the subcutaneous adipose tissue, here the visceral adipose tissue, before, after 4 months, as explained previously. And you have the volumes, and you can see the composition changes and the fat fraction as well. So the main result you need to look at is the fact that at the end of the fasting, there was a higher reduction, minus 35% of visceral adipose tissue, compared to a lower decrease in the subcutaneous adipose tissue. And what was interesting is that the visceral adipose tissue remained below the baseline value 4 months later, while it returned to baseline at 4 months for the subcutaneous components. What is interesting also is the changes for which we do not necessarily for this stage and or the other stage, we don't necessarily have the explanation why the polyunsaturated or saturated fatty acids change in a given direction, and it needs, of course, confirmation in a larger scale. But we observed fatty acid composition change. It's stable at the end of the fasting. You see it's nearly the same value, but after 4 months, there is a delayed increase in saturated fatty acid at 4 months. Okay, so this is a bit interesting here. what's going on, what is the meaning, we don't know, but I think this is something that need to be underlined. Now, if we keep this lower abdomen values in mind, what is interesting is, unlike in the lower abdomen, the saturated fatty acid in subcutaneous and visceral adipose tissue decrease in this upper abdomen, okay? So the behavior of the composition of the different components of fat is not the same, and you cannot just consider one sample as a result of the whole body. We are more complex, and we have seen that over the last 2 days. It means that we deserve and we need more precise, localized analysis, because it does not behave as a single sample. So what it means for us as well, at this stage of the results section, it means that it makes sense for us to keep spending time to look at different locations, because we could have said, okay, this is the same, so we can simplify it and speed up the acquisition. So, so another point I would like to make is we can have maps of the tissue. Potentially, we can, at this type of analysis, I show you what's going on in the whole organ or the whole composition at a given stage, but we can eventually look at difference in different segments if it makes sense. So that's one of the strengths of the analysis. If I want, probably, I don't know how I am in time, a bit late, but I would like to now look at the tides to see, and you can see here maps of the tides with the different kinds of fatty acids, saturated, mono and polyunsaturated fatty acids. So the first point that is important is that we observed a minimal muscle volume decrease. We measure it as -3%, but interestingly, it remains roughly the same, and it's even below. I'm not sure it's statistically important, and I hope you understand that why we don't have tests, because I have just one subject, so I cannot compare it to other subjects. But nevertheless, there is a trend that is interesting to note, and what is interesting also is that there is a decrease in the subcutaneous fat, while the bone marrow fat, that's the next slide, increase, okay? So if we're looking at what happens in the bone marrow fat, keep in mind that bone marrow is actually one of the largest organ in the body, okay? We are usually considering bone marrow as something behind the scene, but it is very important. And what we what we found is that the behavior depends on the location. Again, in the spine, we see a plus 10% increase in the global adiposity, which is in line with other findings, with continuous trends to increase of this fat component. And there are changes as well in the fatty acid composition in the spine marrow. A lot of things happen in the spine bone marrow, whereas in the femoral bone marrow, not much happens, okay? So this, I think we definitely need to target this bone marrow, because a lot of things going on are happening in this bone marrow, and keep in mind that the adiposity in the bone marrow is made of different components. There is that are behind the scope of today's presentation, but there are a lot to be said and analyzed on this adiposity in the bone. Last results is spectroscopy in skeletal muscle using proton. We did not have reliable enough at that time phosphorus results, but I would like to show you very interesting things regarding this changes occurring in the fat in muscle. One thing that I haven't said to you is spectroscopy of the proton can differentiate, and we know that from since the 80s or 90s, that we can differentiate the peak that correspond to the extracellular fat components and the intracellular fat components. So we can differentiate both, and we can look at the kinetics of what's going on in each of these components. So we find an increase of this composition. This is known, and this probably reflects this metabolic switch using lipolytic oxidation. And probably the most exciting, maybe not the most, but a very exciting thing is the carnosine molecule quantification that can be done uniquely with spectroscopy, because you cannot have it from blood because it's very unstable in blood. So you cannot do blood sample with carnosine, but it's a very powerful antioxidant biomarker, and we can show interesting changes of this antioxidant biomarker that also increase after fasting. So quickly, this discussion is, I think we demonstrated that we can enable a detailed quantification of both global and organ-specific composition during such an intervention, and we can more specifically monitor both fat content as well as its different components in many of the organ of interest. Spectroscopy is a unique method to monitor the location of the fat in muscle, and very interestingly, this carnosine is increasing a lot, and it's a molecule dipeptide which is involved in many aspects of the antioxidation against ROS, RNS, anti-age, and advanced lipoxidation end products. So it's a very interesting feature. What are the perspectives? The perspective is, of course, to do a larger study now and to design a study that is well designed to control potential confounding factors. We definitely would like to have a proton phosphorus spectroscopy to quantify the oxidative capabilities, because the same mitochondria is a powerhouse that do the lipoxidation, and so that's typically one specific target that could be of great interest. And we would like to use some new aspect that we develop in the meantime. We can now have a very quick and fast spectroscopy, and we can get images from the spectroscopy. So it's roughly, here the values correspond to the metabolite itself. So you have a phosphocreatine map. The content in each voxels or tissue is actually extractable. And I would like to thank, of course, the Boehringer Clinic and Françoise especially, as well as all the people in the lab that have been involved in the studies. Thank you very much.